Hello and a very warm welcome. Welcome back to this series. In this series, we are framing a strategy on how you can get 300 plus score in botany optional in your main examination. Definitely, it is not an impossible target. Definitely, it is not a very difficult target. You need to have a strategy. You need to have determination to execute this strategy, and you will find yourself scoring 300 plus, and that will change the entire game of the main examination for you. Now, this is a six-part series, and in this series, I am basically trying to cater to the needs of those students who are taking the civil service examination in 2025 and 2026. You could be taking the Indian Forest Service examination as well. The syllabus remains 99% identical. Only just a few topics here and there are different between Forest Service and the Civil Service. And when we discuss the topics, we are taking care of both the examinations. In this particular video, we are going to take a look on paper 2 syllabus of botany. In the previous video, we have already done paper 1 syllabus of botany. We have decoded that syllabus, we have restructured that syllabus, and we have created an order of the topics in which they are going to be best understood by you. Now we are going to basically treat the paper 2 syllabus in very much the similar manner. Here we go, decoding the paper 2 syllabus, and we are going to see syllabus overview of paper 2. Then we are going to see the restructured paper 2 in detail, and finally what are the most important topics of paper 2 which you should be prioritizing at the time of revision. Let's see what are my general observations. So these are the areas of paper 2. Paper 2 is something which is a hot favorite of all the students who take botany, because paper 2 contains such areas which you must have studied during your 12th days if you have done bio in 12th, which you take great interest in if you have prepared for the medical entrance examination, and as such, given the conceptual nature of paper 2 topics like cell bio, genetics, plant physiology and ecology, we end up liking this area so much. But you know what? Every single competitor of yours is also liking these areas equally. Therefore, you need to put a little bit of a strategic work in paper 2 as well if you want to secure a competitive advantage. More about that in the later videos, but these are the 5 areas in paper 2 of botany. First and foremost, you have got cell bio. That is a small and foundational area that will be setting the stage for entire paper 2, because genetics, biotechnology and plant physiology will be depending a lot on your concepts of cell bio. Then we have got genetics, molecular biology and evolutionary biology, one or two topics. Then plant breeding, biotechnology and biostats. Fourth topic is plant biochemistry and plant physiology. Fifth topic is ecology and plant geography. These are the topics. Each one is interesting. Each one is very conceptual. There are limited number of facts that you will have to memorize. Rest of it is all about mastering your concepts. Let's see the restructured syllabus of cell bio. So cell bio starts with techniques of cell bio as a topic. Now this topic is not very clearly defined what all techniques you have to study. But if you look at the previous papers, the techniques of cell bio basically include the electron microscopy, phase contrast microscopy, compound light microscope, and that's pretty much all. They have not asked any other technique of cell bio. Had they listed the name of techniques, perhaps they could have asked more, but because it is not an elaborated topic where the UPSC has given you the list of techniques that it wants you to study, therefore the examiners have set the questions very conservatively only around microscopy within the techniques. They have not gone into any other technique. Therefore, I will recommend you stick to these techniques. One, very few questions are asked, and even if questions are asked, the questions are centered around these few elementary techniques. Then prokaryotic and eukaryotic cells, both their individual accounts as well as a comparative account. And when you are doing eukaryotic cells, you should also be doing the origin of eukaryotic cells, what is the current theory on that, and how, what are the sequences, what are the events which led to the origin of eukaryotic cell. फिर आप आ जाते हो structure and function of these 12 things. First, extracellular matrix. In that context, you will be studying cell wall. Then membranes, cell adhesion structures, both at the membrane and ECM level, chloroplasts, mitochondria, endoplasmic reticulum, and in the older literature, Golgi apparatus of plants were called dictyosome. The syllabus mentions dictyosome, but basically it is one and the same thing. Basically, you have to study Golgi apparatus, then endosomes, lysosomes, peroxisomes, and finally cytoskeleton and microtubules. That part of the cell bio is a structural cell bio. In each one of them, you have to deal with the structure. Also, you have to deal with the function. And when you are talking about a structure, you have to go into ultra structural details. Now, if you see the next part is about nucleus and chromosomes. This is an interesting and important part, both because more number of questions have been asked from the topics which are listed on this slide of cell bio rather than the earlier slide. The earlier slide is more about the structures associated with cell bio, and this slide is more about the nucleus of the eukaryotic cell and the processes of the cell biology. So here we are. We have to study nucleus, then nucleolus, and within the nuclear envelope, you have got nuclear pore complexes. That also you have to study. Then chromatin structure, nucleosome structure, and how the genome is packaged using the chromatin and nucleosome. Then two special types of chromosomes, polytene chromosomes and B chromosomes. These are the supernumerary chromosomes. And if you are preparing for the Forest Service examination, you should also be preparing about Lamb-Rush chromosome. In that syllabus, Lamb-Rush chromosome is also mentioned. After that, all the remaining topics of cell bio are about the processes of cell bio. So UPSC does not give you this kind of reorganized syllabus. That is something I have done so that when you are studying cell bio, you proceed in this fashion. That way you will be able to build better concepts of cell bio. Okay. So now, first process is cell signaling. How does cell signaling happen? The receptors, the signaling molecules, the transduction mechanism, the important types of signaling, all of this. Okay. Questions have been asked. Mitosis and meiosis, highly important, especially meiosis. And within meiosis, the prophase 1 of meiosis. Molecular basis of cell cycle is another very important area that you have to do. And then numerical and structural variations in chromosomes and how it is important. Extremely important. Every alternate year, either in the forest or the civil service, you will find one short note from here. And finally, membrane transport and vesicular transport. This is mentioned earlier in the syllabus, but as a part of cellular process, I have brought it here. So that will end cell bio. If you have got the right books, if you have got the right approach, you should be doing cell bio in not more than 15 odd days with your self-made notes. And then I think this is a repeat. Yes. Anything that we are leaving? No, we are not leaving. And now we come to genetics, molecular biology, and evolutionary biology. Now, genetics part can be divided into two segments. One is transmission genetics, जिसको हम classical genetics भी कहते हैं, ठीक है ना? So there you basically deal with the laws of inheritance, the patterns of inheritance, and what are the factors which affect the inheritance ratios. For example, linkage can affect the inheritance pattern, crossing over can affect the inheritance pattern, then different types of allelic interactions like incomplete dominance, codominance will be affecting the ratio. All of these things have to be studied in transmission genetics, and you also have to deal with cytoplasmic inheritance, sex determination, sex-linked inheritance, so on. Let us see what the topic says. The first topic is a dead one. उस पे question नहीं आता. Development of genetics. That is basically history of genetics. But second to 12 topics are asked. And progressively as you go below from 1 to 12, the topics become more and more important. So gene versus allele concept. For several years, this question has not been touched. It is as good as dead, but you should do it. Then pseudo allele. Hardly questions are asked, but you should do it. Because as you go down, there is an increase in their importance. Okay. Then incomplete dominance, they ask. But you know what? There is a hidden topic here. Before you do incomplete dominance, you need to do Mendelian laws of inheritance. Otherwise, how will you understand incomplete dominance? Then polygenic inheritance. Again, to explain polygenic inheritance, you need to have platform of Mendelian inheritance so that you can basically compare how Mendelian inheritance and polygenic inheritance, they show two very different ratios, and why do they show two very different ratios. And the related phenomenon is something that we know as quantitative genetics and multiple factors. So that is there. We will be seeing it at topic number 4, quantitative genetics, multiple factors. So these are related phenomena, polygenetic inheritance and quantitative genetics. Then incomplete dominance, multiple allelism, these are deviations of Mendelian inheritance. You need to do that. Then we come to the phenomenon of linkage mapping, how do we carry it out and all that. Then you do crossing over, and in crossing over, a small part of crossing over is basically going to involve molecular genetics, that is molecular basis of crossing over. Without that, you won't be doing well in that. Then methods of gene mapping, in this not only linkage mapping, but you are also going to deal with the molecular mapping. Okay, so be ready with that. And then the 11th topic is all about sex determination, sex chromosome, sex linked inheritance, and the molecular basis of sex determination. Finally, we have got cytoplasmic inheritance, cytoplasmic genes, and you are going to deal with the genetics of male sterility. Very, very important. And in this, there is no joke that as you move down this list, the importance of the topics increase. And here you have got the remaining topics of genetics, and these are topics of molecular genetics and evolutionary genetics. From this slide of genetics, the topics which are listed here, more numbers of questions get asked. Okay, more number of questions get asked. So mutations, good number of questions, and inside of the topic also questions are asked, like types of mutations, mutagenesis, and so on. A structure and synthesis of nucleic acid and proteins. That is another way to say that you study DNA replication, you study transcription, and you study translation. Genetic code you should ideally do between transcription and translation. Then regulation of gene expression in prokaryote separately, in eukaryote separately, and certain mechanism of prokaryote like operon based control, attenuation based control are the things that you should be doing separately as important short note. And then an extension of gene regulation is gene silencing. So along with gene regulation, you may like to do this gene regulation in eukaryotes. Then we have got these three topics of evolutionary genetics. So evolutionary genetics will include multi-gene families, then organic evolution, evidence, theories, and mechanism. So certain important theories are to be done, what have been the mechanism of organic evolution, and you have to do the modern synthesis as well. And role of RNA in origin and evolution of life is one of the most important topics that you will be finding. Okay, and then we come to plant breeding, biotechnology, and biostatistics. Now again, what I have done, that I have basically broken the syllabus into thematic areas. So first and foremost, core methods of plant breeding, introduction of varieties, selection, and then hybridization techniques like pedigree, backcross, mass selection, and bulk method. Once you have done that, then certain advanced breeding techniques will be coming, mutation breeding, polyploidy, breeding for male sterility, why do we do, how do we do, etc., heterosis breeding, and use of apomixis in plant breeding. These are advanced breeding topics. बायोटेक्नोलॉजी दो पार्ट में बढ़ता है. फर्स्ट बायोटेक्नोलॉजिकल मेथड्स, बट बिफोर दैट you have to do biotechnological tools. So DNA, RNA probes, southern blotting, how do we do it? How do we carry out DNA fingerprinting? how do we carry out PCR, what are the variants of PCR, and how do we carry out fluorescence in situ hybridization, that is FISH. And when you are doing this, also do the genomic in situ hybridization question has already been asked. And then you have got the biotechnology part. So in this, you do development and use of molecular markers in plant breeding. Then you get to do DNA sequencing. How do we carry out? Don't leave it at only Fred Sanger's method, do Maxim and Gilbert method, do the next gen sequencing also. Genetic engineering, especially with the context of bacterial genetic engineering and plant genetic engineering, and how do we transfer the gene of interest in the plants. Okay. Then transgenic crops kya hote hain? What are the types of transgenic crops we have developed? What have been the methods by which we have developed? And what are the applications of these transgenic crop? That is something you have to do. And what are the biosafety related aspects associated with them? And then you come to biostats. Here, these are the topics. First and foremost, you will be doing statistical measures and analysis where you do a standard deviation, coefficient of variation, and then you do a statistical testing, z-test, t-test, and chi-square test. Then go ahead and do probability and distribution. In distribution, you have to do normal distribution, binomial distribution, and Poisson distribution. So these are the distribution. And then correlation and regression. Biostats is over. Biostats से एक ही short note आता है, but biotech and breeding are pretty important. And after this, what you have got is the biggest area of paper 2, that is plant physiology and biochemistry, highly conceptual. But if you do not do it well, every other competitor of yours will be performing at your level, and that will basically mean that you will be left with no competitive advantage. Therefore, it's strongly recommended that maintain your competitive advantage and go for very nice preparation of this. Topics are as follows: water relation, in which you will be doing ascent of sap, absorption of water, and transpiration. And in this process, you will be doing anti-transpirants also, water potential concept, so on. Then mineral nutrition, how do the plants take the mineral elements? How do they move? What are their mechanisms? And last but not the least, don't forget to do the concept of mineral essentiality. What are the essential elements? Among the essential elements, what are the macronutrients? What are the micronutrients? These are the things you will be expected to do here. Then photosynthesis, photochemical reaction, photophosphorylation, and carbon fixation pathway. Carbon fixation pathway is the Calvin cycle. Photorespiration, you all know, that is photosynthetic carbon oxidation. Then C3 pathway, that is nothing but Calvin cycle. C4 pathway, that is Hatch-Slack cycle, and crassulacean acid metabolism. All of these pathways, how do they compare with each other? What is the relation among them? What is the need for CAM and C4 pathways? And what is the anatomical adaptation associated with C4 pathway? That is the Cranch anatomy. All of this needs to be done. Photosynthesis and respiration are two most important topics. Then how does substance move through the phloem? That is mechanism of phloem transport. And then you come to respiration. What is respiration? What are the steps of cellular respiration? What is aerobic respiration? What is anaerobic respiration? And when you are doing anaerobic respiration, what is fermentation? And when you are doing fermentation, what is acid fermentation? What is alcohol fermentation? Right? What is the reaction? What are the applications? Do that. Then Krebs cycle. After Krebs cycle, you will be doing electron transport chain and oxidative phosphorylation. Then chemiosmotic theory, ATP synthesis. So if there is a year when they do not ask you a question on photosynthesis, they are going to ask you a question on respiration. That's all. Then lipid metabolism. How do the plants synthesize the fatty acids? How do the plants break their fats and oxidize their fatty acids? So synthesis of fatty acid, oxidation of fatty acid, and don't forget to do glyoxylate pathway. That is also asked. Nitrogen fixation by symbiosis, by free living organism, and how does the plant assimilate nitrogen? That is, plant does not keep nitrogen in the ammonia form. Plant will be integrating that ammonia in one or the other organic skeleton. How does the plant do that? Then enzymes kya hote hain? What are the properties? How do they carry out their reaction? How are they regulated? So on and so forth. And then do the coenzymes. And finally, once you have studied photosynthesis and respiration, make sure you are doing energy transfer and energy conservation. So do both the topics here. That will be a very nice way to go about it. But only after you have done photosynthesis and respiration. Otherwise, you will not be able to make a good job, do a good job at energy transfer and energy conservation. Here are your next set of topics. Growth substances, basically the plant hormones, auxins, gibberellins, cytokinin, abscisic acid, ethylene, and brassinosteroid. These six, what is their chemical nature? How are they synthesized? Where are they synthesized? And in synthesis, you don't have to do the synthesis pathway, but what is the name of the pathway? What is the precursor? So on. And then what is the role and what are the applications in agriculture and horticulture? Then you have to do pigments at photoreceptor, phytochrome, cryptochrome, phototropin, and in photosynthesis, you have already done the plastidial pigments, that is chlorophyll, carotenoids. So that will be not required to be done here. वो उधर करना है हमें और यहाँ पे हमें करना है rest of the pigments. And then photoperiodism क्या होता है, कितने तरह के photoperiodic plant होते हैं, what is the mechanism, how does it control flowering. Similarly, vernalization क्या होता है, how it does it show, in what kind of plants does it show, how does it control flowering. And then finally, physiology of flowering. Then you have to do seed physiology, dormancy of seed, what are the factors, what are the types of dormancy, how is it controlled and all. Then storage of seeds, germination of seeds. Then fruit ripening, how fruits ripen up. And finally, you have to do the secondary metabolite and plant movements. Then there are a couple of more topics. Senescence of plants, that is turning old of the plants and how eventually the plant or the plant parts die away. So that has to be done. Then how the plants grow, what are the growth indices, how do we measure the plant growth, how is it happening, क्या हो रहा है, क्या नहीं चल रहा है. And finally, growth movement, something that you have already done in plant movements. And lastly, you have to do four types of plant stresses, that is heat stress, water stress, salinity stress, and metal stress. Big topic, but highly fruitful topic. And once you have done all of this, you are at plant geography and ecology. If you do this part well, it will help you immensely in your general studies also. So पूरे दिल के साथ करना ये topic. So first and foremost, start with what is ecological community, इसकी properties क्या होती हैं, how do we study the community properties, what are the processes happening in the community, so on, and then do succession of plants. After do ecosystems again. What is ecosystem? How do we characterize ecosystem? What are the components of ecosystem? How does the ecosystem run? What is the energy relation in the ecosystem? What kind of a species association we see in ecosystem? So on and so forth. And then what are the major types of ecosystem? Ecological factors within the ecosystem, only the abiotic factors will be coming up for a discussion. Then do concept of biosphere, biomes, and biogeochemical cycle. Four cycles are required to be done: nitrogen cycle, carbon cycle, sulfur cycle, and phosphorus cycle. Do it well, and in the end of every cycle, also do how human interference is kind of ruining these cycles. Then these are 8 topics related to conservation. Biological diversity, biodiversity convention, third topic, and how are we conserving biodiversity. जितना current डाल सकते हो डाल देना. It will help you in both civil service as well as forest service as well as in general studies. Protected area network are established under the Wildlife Protection Act. That is very important. And for our wildlife protection, और यहाँ पे आप नेशनल पार्क, सेंचुरी, कंजर्वेशन रिजर्व, कम्युनिटी रिजर्व के बारे में सीखोगे. एंडेंजर्ड प्लांट क्या है, कौन मेजर करता है, रेड डेटा बुक यहीं पे पढ़ लेना, राइट. एंड देन व्हाट आर द एग्जांपल्स एंड ऑल. IUCN की कैटेगरीज आपको पता होनी चाहिए. यू नीड टू नो साइंटिफिक definition of each category of IUCN. Invasive species you have to do, how do they impact the local biodiversity and so on. And then finally you are going to do the phytogeographical regions of our country. So once you have done this much, ओके, सो कह लो कि इकोलॉजी ऑलमोस्ट कर लिया, लेकिन थोड़े से टॉपिक और रहते हैं। एनवायरमेंटल प्रोटेक्शन में यू हैव टू डू डिफरेंट टाइप्स ऑफ पोल्यूशन, एयर पोल्यूशन, वाटर पोल्यूशन मेनली, and then its control, and also soil pollution, in which solid waste pollution is very important. Phytoremediation, how do we control the pollution by use of plants and all. Environmental impact assessment, that is a management practice whenever we are designing a project. Environmental Protection Act of 1986, what does it say? How has it been able to protect the quality of environment in the country, so on. Concept of sustainable development and global warming and climate change. ये टॉपिक सीधा सीधा एनवायरमेंटल इकोलॉजी को कनेक्ट करता है जनरल स्टडीज में। प्लांट इंडिकेटर, वेरी स्मॉल टॉपिक, क्या होते हैं, क्या क्या पर्पस वो सर्व करते हैं, फुल लॉट ऑफ एग्जांपल्स। देन 10th इज अबाउट फॉरेस्ट्री रिलेटेड टॉपिक्स। फॉरेस्ट क्या होते हैं, कितने टाइप के फॉरेस्ट हमारे कंट्री में है, व्हाट इज द इकोलॉजिकल एंड इकोनॉमिक इंपॉर्टेंस ऑफ फॉरेस्ट, देन हाउ आर वी कैरिंग आउट एफॉरेस्टेशन, डिफॉरेस्टेशन क्या होता है, क्यों हुआ है हमारे कंट्री में, व्हाट इज सोशल फॉरेस्ट्री, एंड द लास्ट टॉपिक इज फार्मर्स राइट एंड इंटेलेक्चुअल प्रॉपर्टी राइट्स। With this, ecology and plant geography will also be over. Now you would be wondering कि वो कौन से सबसे इंपॉर्टेंट टॉपिक हैं जो आपको बिल्कुल से करना चाहिए। सो फर्स्ट थिंग कि इसके पहले कि मैं कुछ भी बोलूं, ऑलवेज रिमेंबर कि यू हैव टू डू 100% ऑफ द टॉपिक। कवरेज के लेवल पे डू नॉट बी एट ऑल सेलेक्टिव। अगर यहां पे सेलेक्टिव हो गए तो सिलेक्शन नहीं होगा, प्लेन एंड सिंपल। सो पूरा कवरेज करना है, एंड देन एट द लेवल ऑफ रिवीजन, ओके, एट द लेवल ऑफ रिवीजन you will be first mastering these areas before mastering other areas. It doesn't mean कि ये करके छोड़ देना है, but master these areas first because they will account for most of the questions which are going to come in paper 2. So first and foremost, genetics, small bio, breeding and biotech. Okay, minus the biostats. That will give you almost 40% of the questions. And plant physio and biochemistry will give you another 35% question. So 75% questions in botany paper 2, they get asked from these two areas, and other two areas like cell bio and ecology will be giving another 25% of the questions. So keep that in mind. पहले पूरा करो and then master these areas like anything. So that brings us to an end of this discussion. Paper 2 decoded. I hope it is sufficiently decoded for you. Now you understand कौन सा एरिया कितना इंपॉर्टेंट है, किस एरिया में किस सीक्वेंस में आपने टॉपिक्स करने हैं। एंड इन द नेक्स्ट वीडियो आई एम एनीवे गोइंग टू टॉक अबाउट द चॉइस ऑफ बुक्स एंड अदर रिसोर्सेज, सो यू विल बी नोइंग कौन सी बुक से करना है, कैसे करना है। सो आई होप you have drawn some value by spending about half an hour time on this video. I am very sure it will give you some direction in your preparation. Please do watch other videos of the series. If this is the first video that you have watched in the series, go back and see other videos in this series also, and make sure that you are watching all other videos which are coming up in this series. Goodbye and good luck everyone. Thank you for being with this series. Goodbye.